To understand plant anatomy, we have to look past the leaves and stems you see on the surface. When you compare a dicot stem to a monocot stem, you see two distinct geometric patterns of vascular tissue. They perform identical functions, but they follow entirely different architectural blueprints. To analyze these blueprints, we first need to identify the building materials. Simple tissues, like parenchyma and collenchyma, consist of only one cell type. Complex tissues, like the xylem and phloem transport networks, are heterogeneous, built from multiple cell types working together. Even the outer layer, the epidermis, adapts its cellular structure to its location. Root hairs are single, uncuticularized cells built for the rapid osmotic absorption of water and minerals. Stem hairs, or trichomes, are usually multicellular and coated in a waxy cuticle to protect the plant and prevent water loss. The epidermal pores are also specialized for their environment. Stomata actively open and close using guard cells to regulate transpiration. Hydathodes, found at the margins of certain leaves, are permanently open pores that facilitate guttation, the loss of liquid water. When building the internal water transport system, plants develop their primary xylem in two distinct stages. This schematic shows the difference. The early forming protoxylem has a narrow lumen, while the later forming metaxylem has a significantly broader diameter. These micro-level structural differences are the specific variables used to distinguish one complex organ from another. Understanding these individual components is the prerequisite for reading the full anatomy of a root or a stem. To identify a root versus a stem under a microscope, check the protoxylem's one-lative position. This split-screen diagram isolates the xylem. In the root on the left, the narrow protoxylem is pushed to the outer periphery. This is the exarch condition. Roots exit to the soil. In the stem on the right, the protoxylem develops inward toward the central pith. This is the endarch condition. This specific direction of xylem maturation, either exarch or endarch, is the definitive indicator of whether you are looking at a root or a stem. Once you have identified a root, you can narrow down the plant type. This technical diagram shows a dicot root on the left, characterized by 2 to 4 xylem bundles and a tiny or absent central pith. Compare that to the monocot root on the right. It has a polyarch configuration, more than 6 vascular bundles, surrounding a massive, well-developed pith. When observing an exarch cross-section, the bundle count and the volume of the pith serve as the primary diagnostic markers for separating dicots from monocots. Identifying stems follows a similar logic. This dicot stem cross-section shows vascular bundles forming a highly organized ring, or eustele, supported by a collenchymatous hypodermis. The monocot stem uses a different arrangement. Vascular bundles are scattered randomly throughout the ground tissue and adictastally. Zooming into a single monocot bundle, the xylem vessels form a distinct Y shape, with an empty protoxylem water cavity at its base. So if you see an unlabeled microscope slide featuring scattered bundles and a water cavity, what is your identification? That is a monocot stem. This spatial arrangement of vascular bundles, whether neatly ringed or widely scattered, dictates both the structural integrity of the stem and its biological classification. Finally, we reach the leaves. This diagram breaks down a dicot, or dorsiventral leaf, on the left. Because these leaves grow horizontally, they differentiate their internal mesophyll, vertical, tightly packed palisade cells at the top for photosynthesis, and loose spongy cells below for gas exchange. Because the upper surface receives direct sunlight, the leaf restricts its stomata primarily to the lower abaxial epidermis to prevent excessive water loss. Monocot or isobilateral leaves generally grow vertically. As shown on the right, the mesophyll cells are uniform and stomata are distributed on both surfaces. They also feature specialized empty bulliform cells that can cause the leaf to roll inward during a drought. The internal anatomy of a leaf is entirely a reaction to its physical orientation to the sun. To pull these rules together, this matrix categorizes the hypodermis, vascular bundle distribution, xylem orientation, and pith size across every major organ. This matrix consolidates the structural logic of the plant body into a single reference point for your exam prep. By understanding these blueprints, you can identify any plant structure through observation rather than memory.